Hi, my name is Alexandra Smith, and I'm an honours student from the Finesse Laboratory of Digestive Physiology and Nutrition. My thesis investigated the sympathetic innervation from the autonomic nervous system to the gastrointestinal tract, specifically the gastric and ileal tissue in rats. Inflammatory bowel disorders, including colitis and Crohn's disease, are on the rise. A report from PricewaterhouseCoopers, or PwC, from 2013 reported approximately 1 in 250 Australians under 40 years old have some form of inflammatory bowel disease. Reports suggest these numbers have already increased since that report and are likely to keep increasing over the next 5 years. These conditions are chronic and often lasting a whole lifetime. The disorders themselves are not life-threatening. However, they're extremely debilitating and greatly affect quality of life. With an aging population with IBD, comorbidities along with IBD that increase with age could greatly affect overall health. Current management of these disorders is poor and often provide temporary relief of symptoms. Gastroparesis is another chronic condition, which is also commonly lifelong. It instead delays the emptying of the stomach, causing vomiting, nausea, and heartburn. Similarly to inflammatory bowel disease, the treatment for gastroparesis is to provide temporary relief from the symptoms of this disease. These conditions are not only a burden to those who experience them, but a great burden economically and generally on the healthcare system. Another condition I'd like to introduce is postoperative ileus. This is not a chronic condition, however, it is poorly understood. It often occurs after abdominal surgery, causing delays in intestinal transit and often results in long inpatient stays post-surgery. As of now, we have no viable cure for gastric disorders, such as gastroparesis, and bowel disorders such as inflammatory bowel disease and postoperative ileus. A new therapeutic technique termed bioelectric neuromodulation is in development to treat these and many other gastrointestinal disorders. Bioelectric neuromodulation is modulation of the electrochemical neural signaling to different parts of the gastrointestinal tract. Often the targets of this neuromodulation are the vagus and sympathetic nerves. These nerves have extrinsic influence over the gastrointestinal tract and modulate the intrinsic enteric nervous system. Clinical trials using bioelectric neuromodulation as a therapeutic technique have had variable success. However, they have been successful in relieving symptoms of gastrointestinal disorders, such as nausea caused by gastroparesis and constipation and fecal incontinence caused by inflammatory bowel disease. Additionally, in animal models, this is a therapeutic approach has been successful for inflammation of the gastrointestinal tract and for the condition postoperative ileus. Sympathetic innervation is known to modulate many homeostatic functions which are disturbed during the dysfunction of the gastrointestinal tract. A downfall of progressing the effectiveness of this therapeutic technique is that sympathetic innervation to the gastrointestinal tract is poorly defined. To better understand how to improve bioneuromodulation and the underlying causes of these diseases, the anatomical organization of the sympathetic nerves to the stomach and ileum need to be further defined, which was an aim of this project. Many collections of soma of neurons which innervate the gastrointestinal tract reside in prevertebral ganglia. One of these ganglia, the coeliac-superior mesenteric ganglia in rats, has been shown to consist almost entirely of sympathetic soma, 99% as quoted by a previous study by McRae et al. This ganglia sits below the celiac artery and surrounds the superior mesenteric artery. Right and left lobes of the celiac ganglia emerge at their ventral surface, and the superior mesenteric ganglia is somewhat merged with the left lobe of the celiac ganglion. Sympathetic innervation to the muscle and mucosal layers of the gastric corpus were investigated, as well as specialized aggregated structures of lymphoid tissue which reside in the mucosal layer of the ileum termed Peyer's patches. Peyer's patches play a major role in the pathogenic defense and homeostasis of the gut and have been coined the immune-sensing structures of the gut. As the Peyer's patches are thought to have a significant role in the pathogenic responses in the gastrointestinal tract, it is thought that defects in inflammation are associated with disorders such as IBD. The muscle layers make up the outer layers of the tract, whilst the mucosal villus layers make up the luminal walls of the tract. To elaborate, the aims of this were to quantify and define the location of sympathetic soma in coeliac-superior mesenteric ganglia in rats. Regions selected for the investigations were gastric corpus for conditions like gastroparesis and the ileal Peyer's patches for diseases like inflammatory bowel disease. Additionally, there was a methodological aim of this thesis to test a new application of an older technique, which was first used in the 1980s called retrobeads. Retrobeads are latex fluorescent microspheres used in the retrograde chasing of neurons. These have been previously only used in the cortex and not in the peripheral nervous system. Unlike other retrograde tracing techniques that have been used in the peripheral nervous system, these beads cannot diffuse across the cell membrane. They have to be vesically transported into the nerve terminals. 12 Sprague-Dawley rats were used in this study. In order to locate sympathetic soma, which innervate these tissues, retrograde labeling was used in the form of injecting fluorescent retrobeads into different parts of the gastric and ileal wall. Four rats had injections into the gastric mucosal layers, 4 into the gastric muscle layers, and 4 into various parts of the ileal Peyer's patch. Retrobeads were injected where they were taken up by the nerve terminals into the tissues they were injected into and retrogradely transported up to the ganglia where the soma of these neurons reside. The coeliac-superior mesenteric ganglia were then removed from the rats and sectioned at 30 microns thickness in a dorsal to ventral direction. The labeled cells were quantified under a fluorescence microscope. Both red and green beads were utilized in this study for double labeling of different parts of the tissues. However, no green beads were seen in section ganglia, which I will discuss later. This image shows a transverse section of an injection site of red beads into the gastric mucosa, whilst this image shows an ileal Peyer's patch, luminal side up of an injection site of red beads into the Peyer's patch under natural light. Now onto results. These graphs show the amount of labeled cells from injections into the gastric corpus muscle. Each bar represents the amount of labeled cells per 30 micron section. The amount of labeled cells increased per experiment as methods were optimized. When analyzing the sections of ganglion, labeled cells were dispersed all over the ganglion. They also seem predominantly found in the celiac ganglion as opposed to the superior mesenteric ganglion, which leads me to my next slide. For the last two experiments on the previous slide, the amount of labeled cells that were seen in the coeliac-superior mesenteric ganglion were able to be differentiated into labeled cells in the celiac ganglion compared to the superior mesenteric ganglion. Anatomical variation is different from animal to animal, but generally the superior mesenteric ganglion makes up about a third of the total volume of the ganglion structure. However, the proportion of labeled cells from the muscle was only found to be 8 and 9%, meaning most of the labeled cells reside in the celiac ganglion. Similarly to my other slide, these graphs show the amount of labeled cells found in each 30 micron section of ganglion, except that these are graphs from labeled cells from retrobead injections into the mucosal layers of the gastric corpus. An observation made when looking at these sections was that many of the labeled cells were on the edge of the ganglion compared to the muscle injection ganglions, which were more evenly distributed across the ganglion. Graphs on the left-hand side of the slide are combined graphs of the amount of red bead labeled cells in each section of the gastric mucosal and gastric muscle injections. The data from each ganglion has been centrally aligned for ease of comparison. The most dorsal section taken that had red bead labeling in it is on the left-hand side of the graph, and the most ventral slice taken with labeling is on the right-hand side of the graph. From these graphs, it can be seen that there is no difference in the distribution of cells in the dorsal half of the ganglion compared to the ventral part. The graph on the right-hand side of the screen is the average amount of labeled cells from the injection into the muscle layers of the gastric corpus compared to the mucosa injection of the gastric corpus, with error bars indicating standard error of the mean. The actual numbers of labeled neurons varied between experiments and was higher in later experiments. Despite this, the difference between muscle and mucosal injections was maintained. From experiments that involved injections into the Peyer's patches, the results were varied. Similarly to the gastric corpus injections, no green beads were observed to be localized in any of the soma of the coeliac-superior mesenteric ganglion, even though, according to experimental methods and analysis of the injection sites, green beads were in fact injected successfully. From examination of the injection sites, it was difficult to see the original site of injection. Regardless, from this data, it can be stated that there is sympathetic innervation to the Peyer's patch of the ileum from the ganglia in rats, more than the control, which was the injection into the peritoneum. The injection into the peritoneum was used as a control to confirm that the beads do not diffuse far from their site of injection. These images illustrate single follicles of the Peyer's patch from sectioned injection sites. The left side of the images are the muscle side, and the right side of the images are the mucosal or villous sides of the ileal tissue. Injection sites are usually identified by elongated sections of clumping. However, it was difficult to see the original site of injection as this clumping appearance appeared in various parts of the Peyer's patch, such as here on the luminal side of the Peyer's patch of the follicle, and here on the muscular side, which is sitting in the submucosal area. However, an interesting observation was found when looking at these images. I observed when there was localization of beads in the follicle, there was also beads localized in the follicle-associated intestinal villi, which are the villi directly next to the follicle. These villi have been suggested to help trap pathogens for immune defense. This may suggest lymphocyte transportation of beads in the mucosa to help mediate the immune response of the follicles of the Peyer's patch. In the section ganglion from the gastric mucosal injections, many cells were found on the edge of the ganglion. These cells may be sympathetic soma, which innervate blood vessels, which is a topographical organization which has been found in guinea pigs. Retrobeads, when compared to other retrograde labeling techniques used previously, such as fast blue, seem to be more specific and accurate in labeling cells. Compared to a previous study using fast blue, the average amount of labeled beads for injections into the gastric wall was 4 times that found in this study in the celiac ganglion. Additionally, when comparing the amount of labeled cells using red beads in the left nodose ganglion compared to the ganglion in this study, a high correlation coefficient was found of 0.85. However, this correlation coefficient is from a small data set, so more comparisons should be done to validate this correlation. A weakness of this study was that no experiments which had green beads injected into tissue layers were labeled. This means colocalization of beads from different innervation sources could not be found, and hence a lot of potential data from this study was lost. The diluent for green beads is different to red beads, which means that they are fundamentally different to red beads in their composition, which may be why this result occurred. In conclusion, there is more sympathetic innervation to the muscle layers than the mucosal layers of the gastric corpus. Substantial numbers of celiac neurons were labeled in the Peyer's patches, indicating high sympathetic innervation to these structures, which may be modulating the inflammation response in the distal gut. Dysfunction in this innervation may be a mechanism underlying inflammatory bowel diseases. The next logical step would be to do an immunohistochemical labeling with neuropeptide Y with labeled beads in the sympathetic ganglion. Neuropeptide Y has been previously shown to label sympathetic fibers, which innervate blood vessels, for vasoconstriction in guinea pigs. Thank you very much for watching and listening to my presentation.